From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 55, recorded on May 5th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you again from LD Home Studios. And apologies up front. I think we ran into some audio snafus from our recording of TWiEVO 54. Word got out to me through a couple sources that it sounded like there was a furnace running or something here. I can put the rumors to rest. So it turns out it was just a work crew digging some trenches to lay Google Fiber cable in our neighborhood. And I had a few windows cracked open, so I think that hit the audio snag there. But we're
all right. Good.
We're good now. I'm happy to report I've battened down the hatches, and it's warming up in the attic here, I'll be honest, but I think we're safe from external sounds.
So you're still at home. You haven't gone in at all to your lab?
Yeah, still at home. So, I do go in probably once a week at this stage just to make sure that the minus 80 freezers are still running, that some of my potted plants are not drying up and wilting, things like that. And then we have a little bit of background stuff essential stuff that we do need to keep going. So some mouse strains and making sure that those breeding schemes and just that they're, alive and doing okay. And that's in cooperation with the great core facilities we have here at the University of Utah. But we're about like a lot of places, that transition is kind of on the menu for better, or I'm worried for worse here in the coming weeks. And so Utah in general has been proposing this system of sort of red status to orange status to yellow to green. And so we've been on red status, as we've been describing currently or recently, and that's transitioning to orange. So this means outside of the university, we were seeing restaurants starting to open kind of in a limited way, trying to do it carefully. But honestly, some of the early signs, I'm a little skeptical that we're going to be able to do this before we get a little farther down the road in terms of having testing, contact tracing, strategies like that in place, really getting folks to wear masks. That's been a surprise to me, is how big of an issue that has become, not just in our community here, but sort of across the country in some of the stories we hear. I just saw the governor of Ohio, for example, just kind of throwing up his hands and saying, well, I guess we won't make masks a requirement. Somehow this really kind of has struck a strange chord of sort of, taking, I think, what to me at least feels like a pretty modest intervention under pretty unusual circumstances to try to, alter our behavior a tiny bit in order to slow this thing down and potentially reopen. But obviously that's hit some real backlash.
That's unfortunate. This should really be a top-down, everybody face masks thing from the federal government. We had a nice letter on TWiV today from a physician in Taiwan who said they've been extraordinarily successful at limiting the virus. They didn't have to shut down anything. They wore face masks extensively. The government ramped up production back in January, and they've been doing a lot of testing in quarantine. So, those two things alone can really work a lot, things that we're not doing, unfortunately. And every state is obviously doing something different, right? Yeah.
It's almost like 50 independent experiments, which seems like
They are experiments. It's exactly right. Because we're going to see in the states, they said, no more face masks. We're going to see what happens. And, I hope it's fine because I don't want people to die.
Of course. Yeah. But no, anyway, so that's where we're standing. And the university is about a week behind. So we're transitioning next week to orange. And so as a laboratory, we're talking about this, in real time, is what do we feel comfortable with? How do we maybe prioritize some things that we could do on a limited basis to sort of get back to the bench? That's obviously, in order to sustain research programs over the long haul, we're looking at that just like folks are looking at businesses, folks are looking at reopening other parts of society. But how do we do this in an organized way where we're taking, in a lot of cases, some really simple steps to make sure that we're doing what we can to mitigate the risks? And so that's kind of the status here. How about there out in New York? What's the latest kind of stuff that you're seeing?
Well, the numbers of cases are dropping. And, the curve, the epi curve of daily cases is clearly going down. And the governor of New York has instituted a 2-week plan. If the hospital admissions drop for 2 weeks, then he will start a graded reopening. Only certain things and not full capacity and so forth. And Columbia is planning to reopen. Columbia Medical School is planning to reopen the labs on May 18th, I think.
Mm-hmm.
So slowly, and upstate it's very different. They've had many fewer cases, and so it's a little bit accelerated. New York City is the hardest hit place in the country, really, because of the density of people, and so you have to be careful. But I've been on a number of calls where business people are trying to figure out, how to get back to work. What do restaurants do? Do we go to takeout for a while? Mm-hmm. No theaters for a while, right? That's a problem. What about construction? That's a big deal. They'd like to go back to construction and so forth. So these things have to be done slowly. And I keep telling people it's not going to happen all at once. You have to grade it back in just to be careful. And if it's overcautious, that's fine. You can't be too careful, right? Yeah.
Well, and it's amazing to me too, is, all of the hard work and sacrifice that we've seen, pretty extraordinary across the country, across the board. How quickly that might be ceded back by going too fast here, and that it sort of takes all that energy to just get that foothold in how quickly it potentially could be lost.
There are ways to mitigate. There are ways to take care of this, and I just hope we do the right thing because there's no need to go back, right?
Yeah, I hear you. So we'll obviously be keeping an eye on that and continuing to comment on that. So moving into TWiEVO's action today, we've got another tasting menu here, Vincent.
I'm hungry again.
3 preprints to consider for our listeners. And we're going to do it this time. So we're going to do 2 coronavirus and 1 non-coronavirus. It is a virus evolution, a virus discovery story that I think is really exciting. It's been sort of lingering in the background, and we keep getting sidetracked. And we'll probably get sidetracked again as usual, but we'll push through this time. It'll be a 3-course episode. So course number 1, this is a preprint that just emerged, I think about a week or 2 ago. The coronavirus proofreading exoribonuclease mediates extensive viral recombination. And this comes from Mark Dennison's group. So Mark at Vanderbilt, long-time coronavirus researcher and physician, pediatrician, if I'm remembering correctly. You can catch up on Mark and his work on a recent TWiEVO coronavirus special that you recorded. Doesn't have an episode number, but it's March 26th was the release date. Mm-hmm. And the work, this preprint, which really caught my eye, I'm excited to kind of run through it. First author is Jennifer Gribble, and this is a collaboration with Andrew Ruth's lab. He is at the University of Texas Medical Branch down in Galveston, Texas. Lots of coronavirus virology going on there. They have the BSL-3 and some BSL. They have BSL-4 as well. Is that right?
That's right. Yeah.
Yep.
Yeah.
So sort of a hotbed of expertise. Andrew, in particular, growing presence in virus evolutionary genomics, which came in with the coronavirus biology that the Dennison lab has been working on for years. I would, even say maybe decades. And so one of the reasons I was excited about this preprint, Vincent, was I was at Vanderbilt a few years ago. So, and that, I think I met Mark on that occasion. And I was hosted by Eric Scarr. He's a microbiologist. I actually just saw Eric is celebrating his lab's 15-year anniversary. We had a really fun visit that he hosted me on. But then meeting Mark, and we had some really fun conversations about bringing genomics to virus biology, to virology, and how you could use some of these emerging techniques, so the long read sequencing, for example, to start to get clues about how the rules of virus evolution, or how do virus populations, how do viruses generate diversity? How do they change over time? What are sort of the concepts that emerge from that kind of mixing that kind of a new analysis? And this is, I think, falls really nicely into the TWiEVO sort of energy, right? Of how do the new technology around sort of genome scale analysis inform our view of evolution. And so we've talked pretty extensively, and I think many of us sort of almost intuitively think about virus evolution. We're sort of comfortable with the idea that viruses mutate really rapidly. That's one of the things that makes them such a challenge from a biomedical standpoint, is that, just as your immune system or your antiviral interventions would get a foothold, then the virus population has changed somehow and they break through, and you have a new epidemic, a new pandemic. The influenza virus and the seasonal pandemics would be a prime example of this. And so we're pretty familiar with the idea that these things are generating rapid point mutations, or reassorting, or as we'll be talking about with this preprint, recombining their genomes to generate diversity. So in the point mutation is a really sort of common theme here, right? That these viruses are just sort of sloppy replicators, that they keep making mistakes. But if you put that sort of population view together with natural selection, that there's some sort of threshold of mistake making that is beneficial or allows these viruses to persist, to sort of sidestep the immune system, or find new hosts that are vulnerable to them. The interesting thing about the coronavirus is, and we've been talking about this now, and for months now, pretty focused on it, is this paradox that they actually proofread. They have the ability, as they're replicating, to be careful in a sense, in terms of not having a ton of point mutations. But then, if that's true, and the virus has a higher fidelity, and this might be a reflection, as we've discussed, of the coronavirus. These are large genomes on the order of 30 kilobases, or 30,000 base letters of RNA. How do you get away with that? If you're actually making relatively faithful copies, and you are limiting virus diversity because of that proofreading activity, how can you then generate enough? And this sort of where this study picks up the trail of recombination, and really highlights sort of what's going on here.
It must be that the proofreading is not perfect, right? Because these, as we mentioned last time, we have about 20 base changes in SARS-CoV-2 since January or so, right?
Yeah, that's right. And actually we'll return to that. So it's not an either or, it's sort of all of the above. And this is a relative, I would say, lower frequency of point mutations. But as you're saying, we're seeing point mutations. We'll, in course 2 of our tasting menu here, we'll return to the point mutations, and some of the excitement, or even maybe over exuberance about thinking about those as they're emerging, and sort of the impact of them will also come into focus. But what's interesting is, point mutations are easy to sort of follow, right? There's, in our DNA sequencing technology, even the approaches that have been around for 40, 50 years, some of the early technology, the Sanger sequencing, it's pretty easy for us to measure point mutations, to basically march along a genome, whether it's a virus genome, a human genome, and say, okay, at this position is this letter, at the next position is this letter. And so I think it follows pretty naturally that, because those are the tools we've had around for so long, is that's where we would look first. And in fact, those are the easiest things to sort of characterize, to put under the magnifying glass, and to pay attention to. I think what we've done is, in doing that, is we've sort of lost maybe some of the plot here about what is some of the key features of how these viruses, and coronaviruses in particular, including SARS-2, how do they diversify? And in a sense, we've maybe kind of have an unbalanced situation where we're under considering the impact of virus recombination. We have talked about that some, and we'll obviously be talking about it more, but with the point mutations that are also sort of in the mix as well. And in fact, these things are sort of, as viruses populations diversify or continue to evolve, these things are happening kind of in the same hand in hand, in a sense. So recombination can move point mutations. Point mutations, there's some ideas that these can even sort of influence breakpoints, or spots where recombination can happen. And so you have these hot spots. We've talked about this some on early episodes of TWiEVO as well. Okay, so actually this paper, very simple experiments, similar to ones we've been talking about. So the old Vero cells are coming back, the vervet monkey cells. Great. The reason these things are great for coronavirus biology is the coronaviruses don't really like to replicate outside of their hosts. And so these cell lines, for some reason, in part because the innate immune system is sort of hobbled or sort of badly decayed in them, allow for the virus to replicate. And so here, very simple experiments to just 2 different coronaviruses now, the MERS coronavirus. So, and remind us, this pandemic or epidemic emerged, when was this and what were the details of that?
MERS emerged around 2013.
Yep. So not that long ago.
2012, sorry, yeah. Not that long ago, right?
Yep. Middle Eastern respiratory, is that right?
Syndrome, yeah. Middle Eastern respiratory syndrome-related coronavirus. So taking off on SARS, but it wasn't SARS, and emerged in the Middle East. Seems to have gone from bats to camels to humans.
Correct. Yep. So that's virus number 1. And then now there are a number of tools or strains that have been brought into the lab over the last 7 years. And then Mark's group also took SARS-CoV-2, some very recently collected strains, obviously, or a strain, and then infected.
That's a good question, Nels. What's a strain and what's an isolate? What's the difference?
Yeah. So, and we'll, another hot button issue here that we'll be talking about some more. So, and thank you. So I should maybe, currently I would say there's only one strain of SARS-CoV-2 that we know of.
I bowed to you, Dr. L. Yes, I'm glad. So you said strain erroneously just now. Before,
I did,
right?
Indeed. And so, so a sampling or an isolate or a sample of SARS-CoV-2 from the one strain that we know about. Yeah, thank you for that'll become increasingly important as the episode continues and as
a strain has actual biological difference, whereas an isolate just has some sequences that we don't know if they're of any difference at all, right?
That's right, and it's a continuum, right? Or and this is part of the confusion and sometimes is that viruses mutate, and so if you grab sort of the marble out of the bag, if there's all these different marbles in there, you'll pull one out and it will be a little bit different than sort of each marble is a little bit unique, and so that's why we would sort of go down the path of starting to identify them uniquely, but you can very quickly sort of go over too far, where now you're thinking, oh, it's a different strain. And then this has all kinds of connotations related to that in terms of, oh, well, if we develop a vaccine for strain A, what about strain B? And so, we need to. And so, anyway, thank you for slowing me down there. So taking a SARS-CoV-2 isolate, again, just very simple infection in a different strain of Vero cells. So these viruses replicate a little bit better depending on the cell line that you use. And then just collecting the genomes so allowing the infections to go for a few days and then collecting the RNA. And so, a very simple experiment. What's new here, what's kind of new to the table, is the analysis pipeline used to analyze at a population level, down to single virus genomes, what are the differences that you can map. And so this is where the collaboration came in with Andrew Roos' lab, and using a tool that he developed called Virema, which is short for virus recombination mapper. Pretty clever. Didn't quite get the viremia in there, but close enough. And so it's a combination here. This is another sort of new advance, I would say, that bringing to the table is the combination of short read or traditional RNA sequencing. And in fact, this is using the Illumina platform, the major engine of genome sequencing these days. And in fact, it's not direct RNA sequencing. You first make a copy, so-called complementary DNA. So you're going from RNA to DNA, a reverse transcription, and in order to apply it to that technology. And then that combined with this Virema mapper, and so taking into account recombination events. So why is that new? So usually when you're mapping these genomes or assemblies of DNA or RNA fragments, you have a template or a construct that a reference genome. In this case, the MERS virus or the SARS-2 virus reference. And then in the short read sequencing, you're cutting these into tiny fragments, millions of them, in some cases now billions of fragments, sequencing short lengths and then assembling them on to the genome. But now if you don't take recombination into account, and by recombination we're talking about you'll be threading along one sequence, nucleic acids, and then you'll recombine or jump onto a different strand. And the source of that, there are several different sources. It could be the virus is nearby. It could be in the context of human. It could be recombination. So the chromosome that was from your mother, the chromosome from your father, these things recombining. This is what gives each offspring sort of some of its uniqueness. This happens in sort of all different flavors and types, including in viruses. And so usually you just take those short reads and you sort of try to put them down onto that assembly. If they don't match, you throw them away because there's enough kind of slop and error in the technology here that you can just punt. You can just say, okay, well, you don't match, so I'm throwing you out. This could be, to use the old saying, throwing out the baby with the bathwater. And there could be some really interesting biology behind what you're throwing out, these recombined fragments that don't quite match. And so that's what virema is trying to do. It's instead of when it sees those mismatches, instead of saying, let's throw that away and just wait for one that matches our assumption about what the virus genome should look like, let's take a closer look at that. And there's, you have to bring in different levels of quality control because it really could be bathwater and not something real or interesting. But that's sort of the step forward here. So first by doing that, and then using some of the complementary technologies that are emerging, these are long read sequencing. And so here, a company that's made a pretty big splash in the last 5 years or so, Oxford Nanopore. And what's kind of fun about this is it's actually direct RNA sequencing. So they've built their technology. So it used to be very similar in the sense that if you wanted to understand RNA, and this could be a transcript, so the messenger RNA from a human cell, or the RNA from an RNA virus, you'd first do that reverse transcription step back to DNA in order to plug in. Just in the last year or two, Oxford Nanopore has brought out this direct RNA sequencing where actually you're threading the RNA itself through the nanopores, literally, in order to make base calls. What is the order of the letters of the RNA as you march along the genome? And so that has some advantages. Some of the bathwater is eliminated because you're going directly to the sequence. And then because it's long read, instead of taking those fragments and trying to reassemble them, you can now read across long stretches. And in some cases, actually, entire coronavirus genomes, the SARS-2 in one go, and then ask, okay, what are the orders of the letters here? And are there any patterns that sort of break what you would expect from your assumption from the reference genome?
So you're actually sequencing the RNA, not a copy of it that's in all the other technologies.
Exactly right, Vincent. And that's a fun step forward. We're excited about this in my lab. So we're thinking about these large DNA viruses, not these large RNA viruses, but their transcripts as they're making RNA as you express the genes that become the virus proteins. What's going on there? It's sort of still a blind spot for some virology, is to really see that directly, in some cases for the first time. Okay, so they use sort of a combination of the short read, but the sort of recombination sensitive mapping. And let's just spend 1 second to define that. So they're calculating what they're calling recombination junction frequencies. So what is that? So what they're basically looking for, and what this virema mapper is doing, is finding, or as it's reading through the sequences, finding deletions that I think are 5 base pairs or more. So you expect this letter to come, and it's like it just skipped 5 before it got back. And so that's diagnostic of non-homologous recombination. It's as if you were on one, you were looking, the genome was going along just fine, but then you spliced in another piece, but you didn't do it perfectly. There's, in that process of recombination, a mistake has happened, and that is that it kind of jammed it together at least 5 base pairs, one direction or another. And so that's the way they're diagnosing this. It's non-homologous, and it's leaving sort of scars of errors in these genomes. And so they'd find a whole sort of treasure trove of what they're calling these subgenomic RNAs, SGM RNAs, and defective viral genomes, basically the errors in sort of splicing these things at the wrong place together non-homologously. And interestingly, a lot of them, as they map it onto the genome, are the very beginning of the coronavirus genome and the very end of it. And in some cases, even not the genes that are coding, but the so-called untranslated regions, kind of the leader sequences that lead up to the genes and the transcription that happens to express those genes into proteins. And so there's all kinds of these sort of highly truncated little pieces of subgenomic RNAs that would comprise defective virus genomes. This thing alone could never persist. It's junk, and it's useless on its own to replicate, independently in a new host. But the idea here is this sort of provides a playground for genetic diversity, that if you have these things all together in collections, and this process of recombination somehow happening at a high level, that then you're bringing this together in new combinations, depending on how these things, splice together in a sense, with a fully intact or a non-defective genome, an effective genome, that now you're generating new diversity, not from just changing a single base, but by sort of swapping these things together over long stretches. Mm-hmm. So the first kind of big, maybe take-home message is just comparing the SARS-2 to the MERS. They saw about 5 times higher recombination junction frequencies in SARS-2. And, don't read too much into that. It's still, the levels are relatively high. I think much higher than maybe you would expect. And there's a lot more of these defective genomes that you would expect. But there could be something going on there. Are there differences in the genes that somehow mediate recombination? Mm-hmm. And the authors get to that point in a little bit. But that would actually, that there's something biological going on there. That is it possible that SARS-2 is really sort of has turned up the volume in a sense on recombination? Whether that has an impact on like how bad the virus is or all of the sort of current virus events that we're grappling with, it doesn't say one way or another. It would just say that these frequencies are higher, starting to define sort of what's going on at the virus population level in a group of viruses that honestly, for all of the attention we're giving to it now, has been sort of a pretty small amount of work that's been done on the basic biology like this. How do these things diversify in populations? So I think what is interesting about this is that it brings up, because you're seeing all these defective genomes sort of enriched at the 5' and 3' ends, it makes you think a little bit about the geography of the genome. Where are the genes arranged? And this is true not only of coronaviruses, large RNA viruses, but also of DNA viruses. So in my lab, we study the pox viruses, and other groups have noted this for a long time, that it's not just sort of a random distribution of genes, that in a lot of cases, the things that are changing the most are out near the ends of the genome. That appears to be the case for coronaviruses. And in fact, if we look at the S gene, which encodes spike, the protein that's getting a lot of attention, as it is one of the sort of the first surface of the virus particle to address a new cell to infect, you notice that's is near the 3' end, and so might be subject to a lot of recombination events. And that's evolutionary virologists, I think, are really interested in some of those features that are starting to emerge as this biology is being defined. So the big question, or the next question that the authors wanted to address, is what is it about the virus that allows for this high level of recombination? And so they turned to a different coronavirus, one that's sort of a distant relative of SARS and of MERS. This is the murine hepatitis virus, a classic that's been studied for many years. In fact, I think you've been talking about this recently on TWiEVO. Is that right?
That's right. We had Susan Weiss on Friday, and she spent most of her career studying mouse hepatitis virus.
Yeah, and so it is a great community of incredibly talented, amazing coronavirus scientists who have, in sort of small numbers relative to the energy that's going in this today, developed these systems over years, including Susan Weiss. One of her former grad student, Stephen Goldstein, is now a postdoc in my lab. And so I've had the Weiss lab on my radar for a long time and been really interested in their work, including with murine hepatitis virus. And so the nice thing about this is because it's not like SARS-2 in terms of it, moving through human populations. It's pretty bad on mouse colonies, though, if I'm remembering right. Is that true?
Yes, that's right. Yeah, you don't want this in your mouse colony. It wreaks havoc. You have to sack them all because you can't get rid of it.
So if you can keep it clean, if you can keep it away from your mouse colony, really useful kind of workhorse for experimentation, because it's less deadly, obviously, than some of the emerging pandemic strains that are of high concern today. And so here, Mark's lab takes advantage of this. They have mutants And they had some clues of where they might look for what's going on with sort of the frequency or the what is it about the virus in terms of its replication process. What are the proteins that might be involved in the recombination process? And so they quickly narrowed in on the non-structural protein 14, NSP14. Can you remind, is that the same as ORF14? Is it the same nomenclature for?
No, it's different.
Different,
the ORFs and the NSPs are different.
Yeah. Classic.
Yeah, some of the proteins are just called ORFs.
Yep.
Because you don't know much about them. And the NSP, non-structural protein, a little bit more. So that's why they get a separate nomenclature. Yeah.
Gotcha. So this is what it does. It's an exonuclease. So this is actually involved in that proofreading, the 3' to 5' exonuclease activity. So basically, as you're replicating a virus genome, if there's a mistake, a mismatch, this exonuclease comes in, it chews back a little bit, and then allows you to try again, to replicate again, to get things correct. That's the nature of proofreading here. What's kind of interesting is that these things are intertwined with other functions, and it's been proposed that recombination is one. There's echoes of this in DNA virology as well. So it's known, for example, that the DNA polymerase of pox viruses also has recombinase activity. And so sort of an interesting theme here, sort of almost dual task proteins or multitasking proteins for not just replication, but also the process, in this case, of recombination. So what they could do is take this mutant mouse virus, which is disabled, has no catalytic activity for that exonuclease. So it's not proofreading anymore. But then the question was, is this also influencing sort of the recombination, the frequency of recombination and the nature of recombination? And so they just take, in comparison, a wild type or a mouse virus, hepatitis virus that doesn't have that mutation, so it's catalytically fine. It has the proofreading, can do the exonuclease activity. And they go into, it's not viro cells, but mouse cells now, do a same, very simple, just replicate it up, get a lot of virus genomes, collect a lot of RNA, and then do the same pipeline. So the short read sequencing with the virema mapping, the recombination mapping, and then again, the long reads to compare that. And what they find is that in the mutant virus, so you take away the exonuclease activity, recombination frequencies went down pretty considerably. And what's also interesting about this is where they map those breakpoints, where recombination is happening in the genome is, so it's lower. You've turned down the volume, but you've also changed the locations. And so the implication of this is that just by messing with the activity of this one gene, that you then change how the population diversifies. There's less diversity in terms of the recombination. And when you do still diversify at those lower numbers, the kinds of breakpoints that happen are skewed in different directions. And so it sort of could change how the virus population actually diversifies over time. And so that's kind of the take home for this paper, is that there's sort of linking this process to this to the recombination process that I think has been maybe underappreciated as an engine for coronavirus evolution.
So it's interesting that this protein, on the one hand, it limits mutagenesis, right? Because it's a big genome, and presumably you need a lower frequency compared to smaller RNAs. Yep. But it's stimulating recombination. So do they have any idea how it does that?
Yeah, I love that. So I think there is work from many years ago, and that's kind of what clued them into using that mutant for this first analysis that gets at some of the mechanism. But it is true. So it's like on the one hand, as this protein's multitasking, you're limiting one sort of flavor of mutations, the point mutation, but then you're increasing or modulating somehow, and potentially even sort of targeting, or as the sequence has evolved, or as the genes have sort of positioned in different places in the genome, the one idea is that this has sort of put this system into a balance, that you're right at this threshold in terms of virus sort of fitness as a population. This is, a little bit speculative still, but that there's been this process kind of in deep coronavirus evolution that has sort of where these patterns emerge. And then we've talked some about this notion of the error catastrophe threshold. So if you, on a point mutation sense, like for a coronavirus, for example, if you treat them with a drug that increases the point mutation rate, or introduce a mutation in the polymerase so that it's even a more sloppy copying process, then all of a sudden you go into this, you pass a threshold, the whole thing sort of breaks down, virus fitness goes down. And this is being explored by some groups as, a vaccine strategy for deploying weakened viruses that are basically are ill prepared to adapt to their hosts. And then our immune system can come back, generate antibodies, and then you have sort of safe and effective vaccines. Is it possible that there's a recombination catastrophe threshold for these large viruses? That's sort of one of the questions I think this work raises.
So it's interesting because in poliovirus, this is a virus that exists very close to the error threshold. And we know this because you can add mutagenic chemicals like ribavirin, and push them over the threshold. They become inactivated. But the polymerase of that virus also has a function to stimulate recombination. And you can change it with a one amino acid change in the RNA polymerase of poliovirus. You can reduce RNA recombination. And when you do that, you lower the error threshold so that the virus is not as resilient. Okay, so the idea is arising there that recombination for polio rescues error catastrophe, which may be a similar thing going on here, except in the case of polio, it's the same protein. Well, I guess the XON would be a separate protein from the polymerase, having both activities, error correction and recombination. But for polio, it's the RNA polymerase, whereas one or two residues control fidelity and a separate residue controls recombination. And you need recombination to make genomes. When there are too many mutations, you can rescue that in an infected cell and get a wild-type virus out. So that's very cool that this is emerging now in RNA viruses, this idea of balancing error catastrophe with existence by recombination. It's very cool.
No, I agree. And it applies sort of across the board to vastly different types of viruses.
Yeah.
But what's interesting, I think, is maybe this pattern that the large viruses, whether they're RNA or DNA, sort of, you have proofreading or high-fidelity polymerases on the one hand, and then high recombination rates, and it might be just slightly flip-flopped for the smaller viruses, and yet they can still sort of work the margins in either direction, depending on the mutation background or the load or the new hosts or the sort of environment that the viruses are replicating in.
The negative strand RNA viruses are generally thought not to recombine, and it would be interesting to apply this long read analysis on infected cells just to confirm that, right?
I agree. And I think the early returns are that there is cryptic or at very low level, some recombination happening, and and this could help to tease that out, to have these new tools in place. Yep. There's, if viruses can do it, usually they are. And then I think what's interesting is the balance of these mutation sort of generating processes and how it might shift depending on the situation. And so bigger picture, the question then becomes, can you modulate the XON activity somehow or target that as a antiviral strategy? And the authors of the paper kind of leave it there as sort of something to think about, given what they're seeing so far. Other questions to consider, though, and I think the authors do a nice job of weighing this, is, this is all in the vervet cells. This is in the viro cells. And does this simple several-day cellular infection sort of replicate what happens in real life and all of the complexity of the camel host, the bat host, or the human host? So those remain, I think, open and important questions. You can always start to compare the patterns that you see in these in vitro cell infection systems to the sampling that you're doing. For example, the thousands of genomes now that are available. And we'll talk about some of the potential issues there, but coming from human patients, that sampling, do some of the patterns or the breakpoints, for example, do we start to see those recapitulated in these more complex infection scenarios? Those are some important questions. And then, same for, so that difference they saw, the 5x sort of boost for SARS-2 versus MERS in the number, the frequency of these recombination breakpoints. Does that represent sort of real virology in the real world, or could it be a difference? The way that they isolated this isolate of this sample versus the way the MERS strain has been maintained over time and how they deploy those, that could actually influence or sort of fool you into thinking there's something going on. And the authors weigh that as well. And so I think a really great preprint in terms of, pointing out, I think, an underappreciated mode of virus evolution, recombination, in addition to point mutations, in addition to things like reassortment, which we're increasingly familiar with for things like influenza virus. It also has, a group that this isn't their first rodeo with coronaviruses.
It's happening a lot these days, isn't it?
Well, and I'm building into our next paper here a little bit. But because of that, there's this depth of knowledge, right? There's this history or experience with the virus, the connection to the literature, the ability to draw on that to sort of balance or polish the arguments, I would say, to make the case or to present the evidence in a way that allows it to be interpreted through a dispassionate scientific lens, for lack of a better term there.
Yeah, I think this is a really nice, refreshing manuscript among all these preprints that have emerged and of dubious merit. This one is solid to the core, really good molecular biology, right?
Yeah. And so, at the same time, it doesn't sort of scream out, okay, here's the cure, or this is what we need to do, or look out. And so it could get sort of lost in the gold rush, in a sense, of all of these papers.
Yeah.
Okay, so speaking of that, we're going to move to our second course now. And so this is...
which may be our last, because I have a 525 out. Okay.
Fair enough. It's good. I need to run. No, it's great. We'll get to this. I guarantee TWiEVO listeners, there are more viruses in the sea, and we'll get there eventually. We'll probably not today. Okay. So this next one, the title of this bioRxiv preprint, and I think this emerged roughly the same time, or actually a little more recently, maybe just in the last week or so. Title is Spike Mutation Pipeline Reveals the Emergence of a More Transmissible Form of SARS-CoV-2. And so the lead author here, this is Betty Korber from Los Alamos National Lab, computational biologist in long-running history, mostly in HIV biology, I would say, and credited, I think, with a lot of contributions in that field over the years, kind of on the computational level. And so, like a lot of labs out there, kind of repurposing a bit. So as the pandemic has emerged, and I think in some ways that all hands on deck there are some good things about that, but we might also lift up some of the limits to that as well. And so this preprint describes a computational analysis pipeline. And this is, we've literally been flooded with a huge swath of virus genomes, coronavirus, SARS-2 genomes, and thousands of them. So we've talked about this at NextStrain. You can see these visualized on these trees, and we've talked about how do we interpret this. There's like so few mutations, and yet it looks like different branches, and some of that, some of the trapdoors almost of getting fooled into thinking there are separate strains here because they're falling on separate branches. But we've got this, now thousands. I think I was just looking at NextStrain. It's something near 5,000 genomes. There's this GISAID repository, genomes getting sampled, SARS-2 from around the world. And so being able to tackle that pipeline is not a trivial thing. And so building analysis pipelines is actually very useful and an important way to start trying to discern or find patterns that might emerge. And of course, and we've talked about this on TWiEVO, we'll continue to, are there mutations that might have functional consequences that emerge during this? I think what we've tried to do is have a balanced conversation about maybe most of the consequential evolution that might happen with SARS-CoV-2 is what happened sort of in the last year or so as it made that spillover event into humans. But of course, now we have all this diversity sort of in real time that people are analyzing. And so having tools to do this is definitely a good pursuit. And so that's what this new pipeline tries to do, or does, and in particular focuses on the S gene, the spike protein, to start to draw out what are there mutations in the spike, given that it is making first contact with host cells as it moves sort of both in an individual infection and then moving between hosts as the virus transmits, and to go through and start to kind of winnow out if there are patterns there. And so the authors propose 14 emerging mutations. And so the way you would start to get a sense that these are emerging is that they're different from the very early sampling that was happening out of the early strains from China. By the way, there's fewer and fewer, if any, or very few, if any, strains that are currently emerging from China. As this thing has progressed and has become more complicated, not only from a scientific perspective, but I'd say from a political perspective, any of the sort of transparency that was happening between many countries is starting to become more limited. And this is an example of that. There's fewer sequences coming from China as they've sort of not only locked down in terms of the virus mitigation, but also in terms of sort of sharing data going forward. Of the comparing those early strains to sort of now contemporary, and contemporary meaning the difference in 2 months or even a month, are there mutations that are emerging? And they say there's 14 already. So we're, maybe an early warning sign in the abstract, which is one of these mutations the authors describe as of being urgent concern, just on the notion that it's becoming dominant. So that meaning that mutation started or was observed very infrequently, if at all, in the sampling out of China in the early days. And then now in some populations or some locations where it's being sampled, and they show some representations of this from different cities, for example, or different countries, where you see this one mutation that is sort of taking over. It's a single point mutation. So we talked about this a little bit on last episode, TWiEVO 54. So the mutation is in the spike protein, the S gene, and it causes a single amino acid, an aspartic acid, or a D, at position 614 of the spike protein. That's just sort of the number of the peptide, the amino acids in a row, changes to a glycine. So D614G is the shorthand for it, meaning that you have a change in the RNA, one letter, which is the change in the amino acid that makes up the spike protein. And there's real interest in this. So there's been kind of whispers that this mutation is, it emerged probably first in Europe and dominates a lot of the strains that have been sampled in New York. Whereas following the sort of virus migration from China to the West Coast, to the Seattle area in particular, only more recently has the G mutation started to sort of emerge or come to more frequency as a later event.
So that means it just could have been introduced later, right there.
Well, that's right. And so that's where the mismatch between the language in an abstract urgent concern versus a single point mutation in a virus starting to become more frequent, that's a pretty big leap to make already. And, to be honest, I haven't read it super closely, but from going through, you see more language like this. So quotations like it's an alarming pace. And, that's true, I guess, so certainly it would be an alarming pace if this was like human evolution that we were tracking, given that this thing is sort of starting to dominate populations over a month. But this really takes kind of virus evolution way out of context, which is mutations are sort of rising and falling all the time. And then the big question becomes, is there a functional consequence to it? There's all kinds of explanations. It could be that this ultimately will be of urgent concern. But again, and we keep coming back to this, I think, as a theme, is where's the evidence, or what is the evidence as something that is as heated as not just sort of the science of coronavirus, of SARS-CoV-2 in particular, but sort of the, public health reaction, the political reaction, all of the sort of major disruptions to life as we know it currently, that really amplifies the volume here. And so as a virus, evolutionary biologist, connecting these words without the evidence is really that to me is of urgent concern, actually, and why I think it's really important that we talk about this and try to weigh the difference between sort of virus mutations as usual from trying to understand what's going on here.
So Nels, the title says a more transmissible form. I mean, it just states it. So what in this manuscript would lead one to conclude that?
Yeah. So I think, again, that's so the pipeline that identifies and tracks or even shows, in the sampling that's been done, and there are these bar graphs where you have, so the D at position 6, the aspartic acid at position 614 of the spike might be an orange bar, and then in blue bars is the G at that position, that mutation. And you can see over time in that sampling and those sort of short thresholds that indeed you might go from a very, sort of a dominant of orange to a dominant of blue. But that's all the information we have right now. We don't know anything about so for that to happen, transmissibility could be the same. It could be that kind of marbles out of the bag process.
Right.
Yeah.
But if you say a virus infects a certain area, and it happens to have a SNP, a single nucleotide polymorphism, that happens to change a non-synonymous SNP, right?
Correct.
And it's there and it's neutral. So it's not giving anything or taking anything away. So that's going to just stay there. And in the people infected from that one person, they're all going to have it. So isn't this a pattern you would see from a neutral change?
It could be. So that's the hard thing to discern here. And so, and just from sort of an intuitive perspective, you might think, okay, well, if it's truly neutral and you could have a D there and the virus would work just fine, or it'd work the same. You could have a G there and it would work just fine. Mm-hmm. It would work the same. That then you might fluctuate back and forth, but selection wouldn't act on it to sort of have one dominate over the other. And so evolutionary biologists have spent decades trying to developing really sophisticated tools to try to discern between that process where you have changes that don't really have a functional impact, or natural selection isn't skewing it one way or another. Because you can have those patterns depending on all of the complexity about how sort of populations grow and expand or migrate or move, that could look very much like natural selection is acting, to try to discern the difference between just that sort of neutral process versus, something's going on here. Something going on, which is in the title, is this is transmitting better. And so if you could link those two things, that's a lot of the energy of evolutionary biology and a lot of the interest here, right? Of course, we want to know, are there mutations that are happening that would explain sort of patterns in these complicated scenarios where the virus is evolving? Again, as we've discussed, I think, just from a sort of frequency argument, probably most of that action has already happened for this virus, and it happened months ago or years ago as it was spilling over. And usually,
in fact, if there were, so there has to be a selection. Well, first of all, your idea that it should flip back and forth if it's truly neutral, but the mutation rate of this virus is quite low. We've only seen 20 bases since December in a 30,000 base genome. So I would say the likelihood of this flipping is pretty low, right?
Yeah, no, that's right. And that's kind of where this, I think the mismatch between the conclusions of this preprint and sort of the information that it presents. So it does build a pipeline. It is measuring, or it is pulling out these patterns from the samples, but it's almost just cataloging. It's not really analyzing that the way, and that's a hard thing to do, to be honest. And you can come at that from different ways. So there are algorithms that might try to discern selection from other sort of non-selective processes, which sort of dominate a lot of population genetics. Or you can also imagine taking, an experimental approach. Okay, we see this pattern, and it generates a hypothesis, and the hypothesis is, okay, this thing is starting to take over, and this point mutation is taking over. Is there a consequence of that? Or you could set up the hypothesis, there is a consequence of that. And then you would try to gain evidence to falsify that, to either support that notion or to falsify or move on to the next hypothesis. There's nothing going on there. And so the authors actually do that. So they teamed up with a crew in the United Kingdom at Sheffield. So it's a hospital where they've actually had patients, and as they've sampled the genomes and maybe just even genotype the spike protein, they have some patients that have the D version, sort of the original aspartic acid in the spike that came out of China, and then they have this new mutation, the glycine, at that same position. And then they make an attempt to correlate. Is there any difference in how these patients are doing? And they find, the authors say and state plainly, we find no significant correlation between the D614G status and hospital status. And so actually, in that case, the evidence that they gathered, if anything, points the other direction from the title, or this makes me less concerned, less urgently concerned, or the alarming pace of this seems less alarming based on that data. They do note, I believe, that so if they do PCR to amplify the virus load from these patients, that they start to see from using the PCR techniques, this approach we've talked about, where the low numbers are good. It's like golf. The lower you get, means the more virus there is. They're able to see those the score is a little bit lower, or there's potentially more virus load in patients with that G mutation. It's very subtle, but then you have to kind of put this into the context. And so, as we've also described and discussed, just because you're pulling out PCR amplified virus doesn't mean you're pulling out real productive virus particles that are transmitting better. And remember, we've just been talking about virus recombination, defective genomes, and subgenomic particles, which could be coming up as part of this analysis. And so, again, we're sort of steps removed from actually measuring what is being suggested here, the transmission and so forth.
I mean, I think it's okay to point out changes that are picked up by your pipeline and then say, we need to do some experiments to address what they're doing. They did this one correlative thing, but you could also imagine going into an animal model, if you had one, where you could measure transmission and seeing if there's an effect. But there's none of that, in which case the alarming words, as you said, the transmission effect in the headline, this is all completely unjustified in my view, and is really a symbol of what this virus has done to some scientists, which is to make them want to get some attention.
Yeah.
Oh, this is amazing. We got to get this out there. And all it's doing is scaring people.
No, I agree. Yeah, this is kind of this rough draft science we're seeing, and so...
Yeah,
and
good title.
Yeah, that's what we did last time, convergent evolution. But we're seeing this where, and I kind of wanted to talk about this point on today's episode, because on the one hand, we're seeing science we're pulling back the curtain and we're seeing how science happens. So you want these tools, and this is great. I mean, this is a step forward, right? The better tools we have to try to understand how this thing is changing, then we might prioritize and do some of those experiments. And in fact, I wouldn't be surprised just that there aren't, half a dozen or maybe even more groups trying to do exactly what you're describing, Vincent, to see if there is something to this mutation. It does catch your eye.
Yeah, but no, I think that's fine. But the
problem is when you get ahead,
it has to be done. But you're right; it's rough draft.
Well, and then and so here there are consequences for that. So, actually, before we get to that, so there's also another thing that is important, I think, to mention, as more and more folks are sort of repurposing their experimental programs or their scientific focus onto the coronavirus, and we'll see how long that persists. But there's also issues, there's artifacts in all of the way you collect genomic data. And so there's a new post on virological.org. I'll put up the link. And we've referred back to this in some of the early conversations we had about the origins of SARS-CoV-2, and still, I think, some of the best ideas that are on there. But this points out issues with SARS-CoV-2 sequencing data, and there are artifacts that come up. So the way both the sequencing technology, mostly Illumina, the short reads, and then the computer programs that assemble these things, there's some hiccups here and there. And so there are, because of that, and because multiple groups sort of use some of the same technology, use the same software to assemble the genomes, there's a set of mutations, and I think there are some in those other 13 that this group also lifts up, that are actually not in the actual viruses themselves. They're just coming up as people are rushing to get this data out, which is a good thing. But again, it's that sort of rough draft version of genome sequences that could then get overinterpreted, and you start to kind of go downstream with this. And that's where you can start to get into some trouble. And so here, unfortunately, there's a story, I think it just came today or yesterday in the LA Times, titled "A Mutant Coronavirus Has Emerged Even More Contagious Than the Original." And it's based completely on this preprint and some Facebook posts from the lead author on it. Again, I think not discussing some of the observations here in a sort of measured way, or that sort of take the evidence at hand into account here. And it's a little more sensationalized. And unfortunately, this is where we run into these sort of, really potentially dangerous scenarios where people are getting freaked out that there's multiple strains. There's not. There's just a handful of mutations, and so we're a little out over our skis, as the old saying goes.
Now, is the The thing you mentioned earlier, that all this probably happened before it got into people or early in this, that's a key here, in that when this virus took off in China, it was already really transmissible, had acquired that property beforehand.
Yeah.
And so you have to put these kinds of theories into perspective. There has to be a selection for better transmission, and there didn't need to be in this case because the virus is already amazingly transmissible. Right. And so that's how natural selection works. Right.
Yeah, that's right. So
I don't see any reason why, even though this is not convincing whatsoever, there are no data to convince you. I still think there's no selection for increased transmission because it's already really, really good at going from person to person.
Yeah. Or another way of saying it is that in order to have selection on even better transmission, it's like turning the volume from 10 to 11. That's really hard to do versus turning it from 1 to 10, which is where most of the action has probably happened here. So another link that we'll put up, sort of pointing out some of these the kind of lack of context here, the selection versus what we might call a bottleneck event, just pulling that random marble from a bag, and it will look unique compared to the whole population, but it's sort of by definition that you just pulled one that you're sort of more attracted to that. So this is from Bill Hanaj, and he posted a nice short blog post on what to look out for and some of the way this analysis, not how it was done, but sort of how it was interpreted and really overinterpreted at this stage.
Yeah, he writes a great quote, but right now there are better ways of fighting the pandemic than worrying about different strains defined by one non-synonymous SNP. And that's great, except strains is not used properly. It should be isolates.
Yeah, agree. Yep. No, we're back in that. There's a lot of levels here. But it matters. So how we discuss this matters because so many eyeballs on this, as there should be. And we really want to be as efficient as possible. And there's going to be ups and downs and mistakes, but I think we need to have these conversations and understand the virology a little bit, a little better, understand the evolution a little better. And honestly, I think this also applies to some of the conversations continuing on about the origin of the virus. So where did it emerge from? This keeps bubbling up. Was it engineered sort of nefariously in a lab? For the most part, that's been debunked, but that still comes back. Part of the reason for that is that, all this confusion that comes out from this sort of avalanche of information and sort of preprints, rough draft science. The other one I think that's even more alarming these days is the notion that this is a lab accident, that the virus is natural, but it was already housed in a lab and then, some workers made some mistakes and then it got loose. There's, again, no evidence for this, but a lot of discussion of it. And again, if anything, good evidence that this is a natural thing. And so how do we sort of rebalance those conversations and put the burden back on evidence? More easily said than done.
I think you nailed it. You said, there's no evidence that this was a lab accident, yet we have evidence that this is a natural virus. And so that's how we work in science. We go from evidence. And so many prominent people are accusing China of this being a lab accident with zero evidence. That's not how science works. Yeah. We have lots of sequence evidence that this came from nature, including recombination and selection of various sequence features. And we talked about this on a TWiEVO some time ago. But it's an accusation. The other side is an accusation. So people want explanations. I have to read you this wonderful quote from TWiEVO. Mm-hmm. So someone sent in a quote from an Atlantic article, and let me scroll down here because this really summarizes what's going on. And we're here because this is a newsworthy event, and so this quote is. It's from this article. It's from an Ed Yong article. He quotes someone else. Mm-hmm. I will put a link. Journalists still think of their job as producing new content, but if your goal is public understanding of COVID-19, one piece of new content after another doesn't get you there. Mm.
It requires a lot of background knowledge to understand the updates, and the news system is terrible at providing that knowledge. Instead, the staccato pulse of reports merely amplifies the wobbliness of the scientific process, turns incremental bits of evidence into game changers, and intensifies the already palpable sense of uncertainty that drives people towards misinformation. So here we have in this paper an incremental bit of evidence that's been turned into a game changer.
Mm-hmm.
Inappropriately. Yeah. I think that really sums up well what's going on here.
Yeah, I agree. And I definitely would point our listeners to this article by Ed Young. I think that is the title something about the confusing times or the confusion around SARS-CoV-2. Really,
yeah, why the coronavirus is so confusing.
Yeah, really well done. And I think it taps into another part of this that really got me thinking, which is, under the circumstances, we all want explanations. We want evidence, and we're looking to science to do that. And yet the scientific process, there's sort of a mismatch here, right? The scientific process doesn't work like that. You don't have sort of authoritative answers right out of the gate. It's this process that goes on. And I think this is an important conversation, is how we communicate as scientists that process, which actually, in a condition like this, doesn't bring much comfort, doesn't bring that sort of authoritative, this is the way to go. And it sort of leaves that hunger. And so how do we have these conversations where it's still an open conversation? We're still open and flexible to new evidence. And in a sense, that undermines the authority, right? And so we're looking for expertise. And yet when we describe the scientific process, what you come back with is, more uncertainty in some cases. And that's just where we are. And that's the confusing times of coronavirus. But how do we try to sidestep or identify that so that our conversations aren't grounded in false authority, that somehow this has to be, this has to be a lab release and, based on nothing. It's almost becomes, just a religious belief as opposed to a scientific belief. And it's really delicate territory.
Yeah, I think it's very hard to explain now. That's what the quote is saying, that science requires a lot of background to really fully understand. And you and I and our colleagues can talk and say, yeah, this doesn't really mean anything, but it would take people a long time to get there. And I'm not sure we're explaining it well either. I'm feeling badly that we get the same questions over and over on TWiEVO, for example. Yeah. We can't simplify it enough. And so people turn to conspiracy theories, right? That's the bottom line.
Yeah. And so I think, as a bigger challenge is, how do we meet each other halfway? How do we, as scientists, try to communicate as openly and simply, but also, to bring and to weigh these things without sounding wishy-washy? On the other hand, how do we, as a general public, start to have enough of a basis to interpret this or to weigh the evidence and have these conversations with folks who've worked on this for 10 years, 20 years, 30 years or more. And so I think that to me feels like currently a big challenge, a big mismatch. It's not one you solve overnight, but I think it's a conversation we need to have.
For sure. Yep, for sure.
All right.
So Nels, we're not going to solve this, but that's a good discussion, I think. Would you like to end up with a couple of picks?
Let's do that. So my pick today is in memoriam. So it's a remembrance of Gordon Lark, written by Mario Capecchi and Elaine Ostrander. It's was just posted on the Genes to Genomes blog at the Genetic Society of America. So Gordon Lark passed away about a month ago. I think it was from cancer. He was 90 years old. But he actually founded the Department of Biology here at the University of Utah during his career. And one of the really great things about it was, sort of from the beginning, the idea was to have a diversified biology department, from ecology to molecular biology, and to sort of mix, not to specialize, a department of molecular biology or a department of virology, but biology, and to have those sometimes messy collisions between different cultures, different fields. And it's really a tribute that department continues to grow, continues with that broad base, and is actually just promoted to a school. But more than that, Gordon Lark is known also for his just recruiting really amazing people, including Mario Capecchi, so that one of the authors, the co-author of this remembrance. And Mario is a colleague of mine now in the human genetics department. and was one of the winners of the Nobel Prize in, I think, 2009 for being able to do recombinant genetic engineering in mouse. It sort of opened up that field of genetics to mouse models. And then Elaine Ostrander from the NIH, who really helped jumpstart the dog genome project and thinking about the artificial selection that we've put on the dogs and to understand the genetic basis behind things like morphological traits. And so Gordon didn't recruit Elaine here, but started several big dog genomics projects in addition to soybean genomics, in addition, again, this sort of Renaissance style of science, and bringing in sort of an incredible roster of really interesting, creative, kind of eccentric scientists, which is sort of marks the University of Utah style. And so anyway, a quick remembrance sort of kind of harkens back to some of the history of the field and how Gordon came up in his career as a phage biologist in sort of the classic group with Delbruck and others, and then how his career sort of went in a different direction here at Utah, and how folks remember him really fondly. And definitely Gordon's emphasis on teaching the next generation of scientists and inspiring the next generation of scientists as sort of a central goal for scientists. So that's my pick of the week.
It's a lovely article written very nicely. And there's this part, it says, it's talking about how the environment in Utah is so magnificent. It is doubtful that any achievements which we have been fortunate to accrue could have been attained in any other environment. Utah was and is a place where people like Gordon could think big, and then bigger still with unfettered creativity. Sounds like you picked a good place, Nels.
Yeah, so that's the voice of Mario Capecchi. That helped attract me here. Yep, and so that's what got Mario here. So Mario Capecchi opened his lab at Harvard, and then Gordon Lark recruited him to Utah on that premise, on that idea, and I see that too. And one of the places why Utah kind of surprises is a fun place, really inspiring place to do science. And so, yeah.
Very good. Very good.
How about you, Vincent? What's your pick of the week?
Quick pick. This was mentioned over on TWiEVO. John Oliver discusses antibody tests for SARS-CoV-2. And he actually does a pretty good job. He's a comedian, I guess, but he does a good job. So if you want to hear him explain it, he'll probably do a better job than I would.
Yeah, so he's on his HBO show last week tonight. I guess you pick about on YouTube as well, which is where this link is.
Yeah, this is on YouTube. Yeah.
Yep. Really cutting through some of the nonsense that was accumulating out there. Really good voice. And so I endorse this pick as well.
All right, that's TWiEVO 55. Boy, Nels, we're going to be at 100 before you know it.
We're doing it. You're like a podcast pusher. It's like a drug pusher, but for podcasts.
You can find the podcast on any podcast player, but the show notes are at microbe.tv/TWiEVO. And we'd like you to subscribe. You get every episode free. It tells us how many people are listening. If you really like what we do, consider supporting us financially. Microbe.tv/contribute. And as always, send us your questions and comments. TWiEVO at microbe.tv. We had a couple here. Maybe we'll get to next time.
Definitely. But
do send them in. Nels Eldy's at cellvolution.org over on Twitter. He's L Earlybird. Good to chat again, Nels.
Yeah, good to catch up a bit, Vincent, and we'll keep it running here.
Yeah, since we're home, we can do more TWiEVOs, and that's cool, right?
Yeah, agree.
Ordinarily, Nels is hard to pin down. He's a busy guy.
We got lots of ground to cover, and some non-coronavirus evolution. I'm starting to stack up some stories here that I'm really excited to share.
All right, let's do that next time. First up, okay?
Sounds good. It's a deal.
I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back soon. Until then, stay curious.